Hi everyone, my name is Edwin Lazo and I'm a scientist here at the National Synchrotron Light Source II at one of our protein crystallography beamlines. And today I'll be talking about automation and robotics accelerating X-ray crystallography at NSLS-II. But before that, let me give you a little background about myself. I was hired at the original National Synchrotron Light Source I at one of their protein crystallography beamlines, where one of my main tasks was to get this little robot that they had up and running, which we did once it shut down. And I'll get back to that little robot in a second. But first, I want to talk to you about robotics. However, before I get into robotics, I want to give you an example of how this type of experiments impact our societies. In 2024, David Baker won the Nobel Prize for using artificial intelligence to design proteins not found in nature to perform certain functions. Now, what his group and collaborators did was they used these special proteins created by AI to neutralize deadly snake venom with great success. Now, why is this important? Well, these AI-designed proteins cut the costs of developing new therapeutics. They speed up their discoveries, and we're able to treat other types of diseases. So this is not confined to just snake venom. Now, what did they actually do here at NSLS-II? Well, they use X-ray crystallography to validate their designs. So what you're seeing here is you're seeing a crystal structure of the enzyme and the snake venom, but you're actually seeing two structures superimposed onto one another. You're seeing the AI structure and you're seeing the experimentally determined crystal structure, and you can see that they superimpose very well. So this shows that they indeed validated their design. Now, in order to get to this answer though, they had to go through a lot of costly samples. Now, how does X-ray crystallography work? Well, you start with purified protein. You crystallize that protein, and then you take these protein crystals and you expose them to X-rays. And that's actually what they do here at NSLS-II at our protein crystallography beamlines. Now, once you expose them to X-rays, you get this diffraction pattern, which essentially are a bunch of spots. And once you have these spots, you can use math in order to determine the shape of the protein. And you end up with something that we call an electron density map. Once we have this shape, you're able to fit the amino acids that make up the protein. And you end up with a 3D structure of this protein, an atomic model, which essentially is the shape of the protein. Now, why is this important? Well, if you take protein A, for example, you know that it needs to have this, it has this pointy shape. Well, in order for another protein to interact with protein A, you need something that will fit into that point of protein A. And when they interact, they induce a biological reaction. So in essence, once we know the structure of a protein, we have a better understanding of its function. Now, why do we want to do this? Well, as I mentioned earlier, we had to go through a lot of samples in order to get our answer that we were trying to get to when we were trying to validate if the structure that the AI model produced was indeed valid. The other motivation is that we want to be able to take advantage of new technologies. For example, the brightness of NSLS-II. Also, every year, computers become more powerful, so we want to take advantage of this new computing power. And also, detectors are getting faster every year. Also, we want to make efficient use of our beam time. We want to be able to run 24/7s, but humans get sleepy. And we also want to offer different types of beamline access modes, which I will get to in a minute. So here, you're going to see a video of the robot in action in our automated workflow. So what you're going to see is that the robot is going to mount a sample. It's then going to center the sample to the beam, which is that blue crosshair that you see. And then once it centers it to that crosshair, It will then do a raster grid that will scan the protein crystal, which is that rod-shaped item that you see there, that rod-shaped object, and it's going to try to determine the best area to collect on. Once it finds that best area, it will collect on that area. And once it's done, the robot will then proceed to unmount the sample and mount another one. Over the years, this has been in operation since March 2017, and over the years we have improved this workflow. So we introduced AI loop censoring, AI-powered rasters. We also incorporated remote controls, not only for the robot, but also for the door. Now the door is a container that has liquid nitrogen that holds our samples. Our samples need to be in cryogenic conditions in order to mitigate radiation damage, just like how we put sunscreen on our skin to protect us from the sun. Also, we worked very hard for the robot to be reliable. So for every 2,000 samples, you would lose 0.8 samples. We incorporated alarms that alert our floor coordinators that we call flocos in case of any faults during overnight operations, and they're trained to recover. We also have sensors that ensure that our samples are kept safe, like a liquid nitrogen level sensor in our door. And we also have equipped our system with sample tracking, such as machine vision. Now, back to access modes. Back in the day, in X6A, before I got that little robot to work, researchers would come in person and they would bring their samples on site to conduct the experiment. However, with all this new modern technology that we have now, we want to take advantage of these robots and how fast they can go. So we introduce remote. And we have two types of remote methods that you can use these beamlines with. The first is what we call classic, where researchers mail their samples in and they control the experiment remotely. So in essence, you can control your experiment right from your bedroom. Now, however, what we discovered was that not everybody was able to get through all their samples because they did not have enough time on the beam. So we introduced a new mode called hybrid, where you would mail your samples just like you do in classic mode, but this time we collect your data automatically using our automated workflow the night before. Then you get a chance to go back and see which samples need human attention. We also have standby mode, where researchers mail in their samples and we collect on those samples using our automated workflow without human involvement. Here you see a picture of results from standby. So on your left, you're going to see protein crystals, and these are the crystals that were collected using our fully automated workflow. And on the right, you see the results. What they were trying to do here is they wanted to see if their drug bounded to their drug target, meaning if their drug bounded to a protein. And it did. So you see the green shape there, that's a drug, and the blue is the protein, and you can clearly see that it's bound to it. This is great significance because this was all done in fully automated mode, meaning no humans were involved in this data collection. These new modes have allowed us to increase our throughput and have allowed us to accommodate researchers from the Advanced Photon Source, which is another synchrotron that has been shut down for a year in order to go through some upgrades. So if you look at this chart, you could see that standby, we went from around 5,500 samples to now 9,000 samples per cycle. And a cycle is a third of the year. So we saw roughly 3,500 sample increase when we introduced this new mode. However, during the APS shutdown, we saw more samples come through, and then what we realized is that we were topping out at around 11,000 samples. But what we noticed as we were supporting our colleagues is that they were not able to get through all their samples. So that's when we introduced hybrid, and if you can see hybrid mode, every sample that comes through our beamlines are measured, and you can see that with the bump up there, where we're now seeing about 13,000 samples per cycle. Now, The blue bars here, they represent proprietary samples. So we also have companies that come and use our facilities because this technique can also be used for drug discovery as well. In order to handle all these samples, we introduce industry standards. So we have the booking app if you want to self-schedule your beam time. We have Microsoft Forms if you want to request standby mode. And all of our sample holders that we call pucks have been barcoded because when we load them into the robot door, we scan them in. And when we scan them in, the samples are automatically linked to our researchers and the experimental parameters which they want to collect with. We're also working on automating more aspects of our operations. So we just recently acquired a crystallization robot that will allow us to generate more samples faster and using less protein. We've developed a center of rotation pen that allows us to align the beamline automatically, and we're working on room temperature mounting. Currently, if you want to do room temperature experiments, you need to come on site and you need to mount these samples by hand. And with that, I would like to thank the team from the Center for Biomolecular Structure that's led by Sean McSweeney. I would like to thank our team members from the Data Science and Systems Integration Division and also the Accelerator Division, where our floor coordinators help us with our overnight operations. Thank you for watching.